The smallpox and polio vaccines contain complete viral pathogens, all their proteins as well as their genes. This approach to produce vaccines against bacterial pathogens is also possible. However, bacteria compared to viruses are pretty big, with large DNA genomes that code for thousands of proteins. Let's take whooping cough, for example. This serious respiratory disease is caused by the bacterium Bordetella pertussis, which is made of some 2,000 proteins. A whole cell vaccine has been produced by killing the bacterium with formaldehyde, in the same way Salk prepared his inactivated polio vaccine. The whole cell pertussis vaccine has saved and continues to save millions of lives around the world. However, sometimes young kids have strong, annoying reactions to this whole cell vaccine, developing an inflammatory response, redness, swelling, severe pain, and fever, which, as it turns out, can all be avoided. Scientists systematically studied the pathogen and mapped out the specific proteins that directly mediate the disease process. These can be used to produce a subunit vaccine made exclusively of only these few but most relevant proteins. It turns out that only 5 out of 2,000 proteins of the pertussis bacterium are enough to train our immune system and provide protection against whooping cough. And, as anticipated, this subunit vaccine has markedly fewer and less severe side effects. This approach of generating subunit vaccines has many advantages. First of all, it focuses our immune system to the most sensitive and critical targets of the pathogen. It reduces unnecessary numbers of proteins that do not provide any protective advantage, yet contribute to unnecessary inflammation. Subunit vaccines themselves are never infectious and thus are especially safe. This approach of subunit vaccines has also been applied for toxic proteins, toxins that infectious bacteria produce. The 2 most famous bacterial toxins for which vaccines are administered routinely are tetanus and diphtheria toxins. These vaccines contain tetanus and diphtheria toxoids. These are the bacterial toxins inactivated with formaldehyde. So, think about it. The triple vaccine, DTaP, given to protect our kids against diphtheria, tetanus, and whooping cough, doesn't contain even one infectious agent, and no more than a handful of harmless proteins. As it turns out, subunit vaccines can be very effective for some viruses as well. When we reviewed the replication cycles of viruses, we learned how cells are only susceptible to infection if they present the cell surface viral receptors. Thus, scientists have developed subunit vaccines that contain only one protein of the virus, precisely that protein that is crucial for receptor recognition and binding to mediate entry. A case in point is the hepatitis B vaccine. The vaccine contains just one spike surface protein of the virus, a relatively short protein of about 200 amino acids long. Moreover, the surface antigen is produced artificially in Baker's yeast cells. So the vaccine has no trace of viral DNA, no unnecessary proteins or lipids, yet affords protection by training and focusing our immune system to effectively inactivate and neutralize the viral spike protein. The most recent subunit vaccine used today protects women and men from cancer. Human papillomavirus, HPV, is a naked, double-stranded DNA virus whose genome codes for 8 proteins. One of these, designated L1, which makes up a major part of the viral capsid. This single protein produced in yeast constitutes the entire HPV vaccine. No DNA, no extra proteins. Finally, when the pathogen's antigens are not proteins, subunit vaccines can actually be better than nature. You recall that bacteria are sometimes slippery and hard for macrophages to engulf? Antibodies directed to the slippery outer surface of these bacteria enhance macrophages' eating efficiency. The problem is that the slippery coat of bacteria is sometimes made of polysaccharides, complex sugars. Here is where scientific ingenuity has paid off. Whereas the natural bacteria are somewhat resistant to our immune system, we can outwit nature and engineer their coat to become improved, excellent targets for B cells. If we chemically attach to the sugars some proteins as well, the sugar antigens become remarkably more appetizing for B cells. These artificially mixed antigens, conjugates of protein bound to sugar, are able to stimulate the production of antibodies to sugars and provide efficient protection against the otherwise slippery bacteria. This is exactly how the vaccines against Haemophilus influenza and Streptococcus pneumonia are produced. The vaccines contain the capsular polysaccharides, conjugated with particularly tasty proteins that together stimulate B cells to generate highly effective antibodies that, when the time comes, enhance the ability of macrophages to eliminate these bacteria.